Good morning, students. I am Anjas Varghese, lecturer of Immanuel College of all of you. Today we are going to discuss about clinical study designs. What do you mean by clinical study designs? Introduction. Clinical study design is the formulations of trials and experiments as well as observational studies in medical, clinical and other types of research. Example, epidemiology can involve human beings. The goal of a clinical study is to assess the safety of or other mechanism of actions of an investigational medical product or procedure or new drug or device that is in development but potentially not yet approved by a health authority. It can also be used to investigate a drug devices or procedure that has already been approved but still is needed for further investigations, topically with respect to long-term effect or cost effectiveness. That is the introduction part. Then study design can be descriptive and analytical. In descriptive observational studies, it is an epidemiological studies which is not structured formally as an analytical or experimental studies. Descriptive studies do not use control groups. They are conducted to describe or summarize data and are helpful in generating hypotheses. Such observational studies can help to describe a situations like occurrence of ADR in a patient's. Distributed observational studies may be case reported and case series. Case reported and case series. In case reporters, a case reported is the presentations of a careful, detailed reported experience of a patient. It focuses on an individual patient who is on a drug use but experience a particular outcomes, generally an advice. They provide answer in identification of a new diseases or advice effect of an expert's drug treatment. Case reporters are hypothalamus genetic report. They begin with evidence that support a hypothesis about drug effect to be tested with more effective and targeted study results. Case reported are more useful when the diseases is uncommon and when it is exclusively cased by a single kind of expert. The single generated from cases report may help for further investigations of studies. Example 1. The case reported of hypertrichosis with the use of mini for treatment of hypertension helped for developing Minocre as a topical preparation for bad acid. Example 2. Cases reported indicating that the primarily likely human target acted as a single generation for confirming that tintonin could produce the system normalized. Advantages. They serve as an altering mechanisms for clinical investigations and other that a drug or group of drugs could produce a given effect. It promotes clinics to awareness of the potential problems and to report other such outcomes. They may directly lead to hypothesis generation that can guide a research. Disadvantages. Case reported are the weakest form of evidence of calculations. It is very rare that a case reported can be used to make a statement about calculations. Then case series. What do you mean by case series? A set of sequential cases report make a case series. Cases series are collections of patients, all of whom have a single exposure and whose clinical outcomes are then evaluated and described. The cases series studies are useful in qualifying the incidence of an adverse reactions and are generally used for the study of newly licensed or marked drugs. The greater the number of common experience, the stronger will be the signal to support the conclusions. Case series does not have a formal triple protocol but helps in learning our knowledge on the associated with the medicines and help to confirm the areas. Example, case series of liver and bowel abnormality of thyroid exposure. Advantages: they can be useful for quantifying the incidence of an adverse reactions. They can be useful for beginning certain that any particular adverse effect of combination dose not occur when observed in a population which is larger than that studied prior to drug marketing. Disadvantage: they lack comparison group. In this absence of a control group, one cannot be certain which features in the descriptions of patients are unique to the explorers or outcome. Analytical. Then second one is analytical observation study. The analytical studies compare a responsible group with the control group and are usually designed as a hypothesis testing studies. Primary goal of the studies is to establish a relationship between a risk factors and on outcome. Analytical studies may be observational, curious, experimental, and experimentals. Then first one, observational studies. What do you mean by observational studies? There are also known as non-experimental studies. They does not involve interventions. The patients are not assigned to treatment by the investigator. The investigator observes the occurrence of the conditions and the diseases in the population groups. The patients who receive medication from conventional settings of medical care are selected as a subject for the studies, often more practical and feasible to conduct. Often there is a little control over the study situations. Then cohort studies. What do you mean by cohort studies? A cohort means group. Hence, a cohort study is a study of groups of patients having some common drugs exposed to ingestion. A cohort study assembles a group of persons without the diseases or inserted at the one set of studies. Then the explorers of status of person is found out and some cohort is followed over the time to determine the development of diseases affect the exported non-exported. Advisors gather data recording sequence of even can access in casualty, examine multiple outcomes for a given explorers, good for investigation of rare explorers, can calculate the rates of diseases in explorers and unexposed individual over time, incident and disadvantage. Large number of subjects are required to study rare explorers. Cohort studies can be progressive and repository cohort, which is common characteristic of the cohort studies to start off with the reference population, some who have certain characteristics exposed with other who do not have this characteristics unexposed. Both groups should at the outset the study be free from the conditions under consideration. Both these groups are then actively observed over a specified period to find out the risk each group has of developing the conditions. Developing in these conditions. Developing the condition. Many progressed cohort study participants risk factors present expressed and not expressed. Then future evaluation of an outcome or not outcome. Not explained of the same outcome and outcome. Beginning studies and end studies. Flowchart. And disadvantage may be expensive to conduct, may require long distribution for follow-up. Maintaining a follow-up may be difficult. Then was to follow up the withdrawals. Then prospective cohort studies. In this study, the investigator record all the expected data from medical records and or computer database that allow him to reconstruct the data pertaining to explorers of cohort to suspend risk practice and the subsequent consequence of patient. substitute recall basis for information based less control over variables. Retrospective cohort studies flowchart. Then exported subject and non-exported subjects. Subject with the conditions and subject without the conditions. Case control studies. A case control studies is designed primarily to establish the cases of the diseases by investigation association between exposures, risk factors and occurrence of the disease. It is a type of theoretical studies based on exposure histories of cases and controls methodology. The study assembles a group of patients who have a common diseases of interest. These are considered as cases. The control group involved the people who are respected of the underlying populations from which the cases come but do not have the disease. The background of the case control study is to compare the prevalence of exposures between the cases and the control. Thereby, the study uses a compression group to establish the extent of association between the exposures and the diseases. Example, two groups patients A and B each undergoing medical treatment for management of acute pain in a recent arteries as treated and selected. A group A patient cases was found to be suffering from peptic ulcer, whereas the group control did not have peptic ulcer disease. Here, cases control to study can be conducted or find out the extended association with the non-steroidal anti-inflammatory drugs used for the management of rheumatoid arthritis. To case of peptic ulcer, the patient by screening the medication patterns of patients, both the group used the management of pain in arthritis patients. Maybe the study can be used to provide the hypothesis that chronic and persistent non-steroidal anti-inflammatory drugs uses in arthritis patients can be one of the potential significant risk factors to develop peptic ulcers. Then designs of case control studies, exposed and non-exposed cases and controls populations. Advanced can obtain finding quickly, can often be undertaken with minimal finding, fitting for investigations rare diseases, can study multiple exposures, generally require few study subjects, cannot generate instant data subject to selections and information also bases difficulty of record keeping in either inadequate or unreliable selections of controls to be difficult. Then cross-section studies. Cross-sectional studies may be considered as all it is a non-synchronous studies. This type of studies are conducted at a specific point of time through a sugarless database analysis or medication chart reviews. They help the collections of data on across sections of populations, which may covers the whole population or a sample of it. Cross-section studies assess both the exposures and outcome currently. Therefore, it is a good for prevalence research. In advance, no waiting for outcome to occur. This makes them fast and expensive. There is no less of the progress. Difficulty of establishing causal relationship from data collected in a cross-section time frame that is now directed estimated of risks. There are also impractical for the studies of rare diseases if the design involves collecting data on a sample of individual from general populations. Then cross-sectional studies, population, track-like populations, exposed, unexposed, then developing outcomes, node outcomes. This is a flow chart of the cross-sectional studies. Experimental studies, also known as the investigational studies, where investigating diseases who will get the explorers and who will not. So under direct control of the investigator, unlike other type prospective studies where explorers is not dedicated by the investigator, aims to experiment to provide a scientific proof of ecological factor, to provide a method of measuring the effectiveness and use of healthcare services. And experimental studies may be classified as randomized control and non-randomized control studies and flow chart types. Then randomized control studies. Randomization is a statistical procedure by which participants are allotted into experimental and control group to receive. Randomization will eliminate the base and the follow for benefit. Steps drawing up the protocol, sample selection from target population, make necessary equation, randomization, interventions, follow up, then assessment of outcomes. And randomization of clinical trial designs. Then methods of simple randomization, group randomizations, and stratified randomization. Simple randomization means it is a rarely used computer generated random number of tablets. Block randomization used to ensure the number of participants assigned to each group is equally distributed. Used commonly in a small trials. Stratified randomization used to ensure that important baseline variables are more evenly distributed between groups than exchanges along a assurance. Advantage, ideal and most definitive studies the effective of drug. Randomized help to minimize the changes of base element selection space. Then this is a highly expensive ethical problem conducting these studies. Then trial which test effectiveness are large and more expensive. Some research cannot be ethically performed in a artist. In non-randomization, also known as a Curie experimental designs. Non-randomized following uncontrolled trials, natural experiment before and after compression studies with control and without control. Uncontrolled trials means there are no compression group. Evaluating whether a specific therapy appears to have any value in a particular disease, to determine an appropriate dose, to investigate adverse reactions. This is including historical controls. Advantage, when the disease is uniformly fatal and no drug becomes available. The natural experimentals. When experiment studies are post in humans, natural circumstances that many can experiment are identical. And before the competitions. In absence of control group, result of compressions may be misleading. Alternative is to utilize the natural control group that is provided by a natural circulator test. An example, each of the seed frequency in the distributed adapted and compared with another distributed forms. Thank you.